All right, we're moving on through the mini lectures. Here's another mini lecture that goes all the way back. We're moving backwards if you're listening to these in order. This is lecture 5G, and we're going to take a quick detour back to the pentose phosphate pathway. This is its own little topic, but it's always put by the book with glycolysis, although it's really anabolism. And so that's one of the reasons why I want to separate it out as a topic. It's its own little topic, but it's another thing you can do with glucose. Remember that we had that diagram that showed you could take glucose, you can turn it into energy, you can burn it, right? Or you can turn it into other things. You can turn it into, citric acid cycle intermediates, I suppose, although the net of that might have been. You can transform it, you can store it as glycogen. That's another thing we said. We had 4 arrows going out of it. One of the arrows is that you can turn it into ribose. And it's just an interesting thing because you need a lot of ribose if you're dividing a cell. You need a lot of DNA, and each DNA base requires a ribose. So rather than forcing you to eat all this ribose, which is a possibility, but wouldn't it be better to take your main sugar, glucose, and turn it into ribose? Great. There's one little problem. You have a 6-carbon sugar that you need to turn into a 5-carbon sugar. Now you could just say decarboxylate it, but number one, that's hard. It requires breaking off a CC bond. Number 2, that's inefficient. You're taking this carbon that you could be using for energy, and you are just chopping it off is not good enough. So the overall reaction that you want to do is you want to turn your glucose into activated ribose because you want to use it to build. You want to turn it into a ribose that is phosphorylated. And that is what the pentose phosphate pathway does. That's what the phosphate is doing. Pentose means 5 carbons. Phosphate means you're putting a phosphate on it, so it's activated and ready to build nucleotides. Okay. The other thing, if you look at the difference between the two, if you're putting a phosphate on it and turning it from glucose into ribose, you also are producing electrons. And you put those electrons on NADPH rather than NADH. Remember, the structure of these is exactly the same. It's got a little phosphate tag. But that phosphate tag means that I usually want to use these electrons for building rather than burning. One of the authors, Nick Lane, actually said NADPH, the P stands for power. These are powerful electrons I want to use for building things because that's what I want to use my power for. Okay, so it's found in many anabolic pathways. In fact, it's one of the ways you can tell that it's an anabolic pathway if it's using NADPH to build stuff. So fat synthesizing tissues will use it. So if you have, we talked about lactose being put together, milk has a lot of fat in it as well. So the same places that make lactose will also make lots of fats, and they will have NADPH that's being used to build those fats up. Liver does this, and of course fatty tissue will also have fat generating ability. So ribose 5-phosphate is most often used for nucleic acid synthesis. The quicker you're growing, the more you need it. And so you might see that there is a cancer connection here as well. So here's the figures, and I want to put this in its own lecture because I want you to approach these figures differently. I am not going to ask you what the names of these things are. I am going to ask you what the products of the whole pathway are. So take a quick look at this and just see that you're making 2 NADPHs for each glucose, and then it makes ribose. And then it can actually be turned back, the ribulose, can be turned back into glucose. But the main thing here is glucose 6-phosphate on the top, ribose 5-phosphate on the bottom, and you have CO2 leaving at some point, but you're capturing a lot of that energy by putting the electrons that are leaving the molecule onto NADPH. So this is a way not just to get ribose, but to get NADPH. And in fact, there is a way of running it. Look at the arrow on the left. Glucose 6-phosphate can go down to ribulose. You can lose the carbon. You can put the electrons in the box. And then, if you don't need ribose, you can actually take the ribulose and put it back up to glucose again. This is what you can do if you need NADPH. So you can turn glucose 6-phosphate into NADPH by doing this processing and by going from the transaldolase to the transketolase. Transaldolases and transketolases. Basically, notice they don't even give you the names of those enzymes. That's a big clue. But just know that you can do this. You can actually have a form that makes ribose, or you can just run it on the mode where it just makes NADPH. This is useful for two different purposes. That's one of the reasons why it's useful. The only thing I want you to see in the intermediates, you can look at the actual biochemistry of this, but there's no time for that. I don't want you to necessarily even know. The only thing that you might be able to have in your head, if you're producing NADPH, you are dehydrogenating something, right? So you have two dehydrogenases, one for each of the NADPH producing steps. The only other thing you need to know is that it goes through a lactone intermediate. Okay, so you take the glucose and you bring it through a lactone, and a lactone just means that you have an oxidized glucose. You are oxidizing it because you're taking the electrons off of it, right? You're oxidizing it, and you oxidize it from a regular glucose to a lactone, which is an ester linkage. And if you can see that that is more oxidized, then you rearrange it, you oxidize it again, pull the electrons off, and the CO2 falls off as well. Then you end up with ribose 5-phosphate, and that's all you need to know. I am not going to ask you the names of these. I'm not going to ask you the specific structures, but I want you to have this in a 5-minute sort of snippet in your head. The overall reaction is what's really important. And it's basically glucose 6-phosphate gets turned into ribose 5-phosphate, and CO2 comes off, 2 electron pairs come off, 2 NADPHs. And so a lot of times the cell will need the NADPH more than it needs more ribose. In that case, it will actually run it as a cycle. It will make ribulose, turn that back into glucose, make more ribulose, basically strip the electrons off of the glucose. Use the glucose to make NADPH rather than to make ribose. But that's actually an interesting biochemical question, because how do you put carbon back on? If cutting off the carbon, it can be inefficient. How much more putting back the carbon back on? So the cell has a really cool solution for this. It basically has a complicated pathway that takes 6 pentoses, 6 ribuloses, and turns them into 5 glucoses. Realize that the atoms all work out that way. There's 30 atoms on each side. But if you think about it, how can you do the organic chemistry to take those 6 pentoses and to turn them into 5 hexoses step by step, so each step makes sense? Well, you don't have to figure it out because the cell has figured it out. And here is the, after you do the oxidative reactions, you go from glucose, the blue box, to ribose, which is the red box on the left. And now you're trying to get back from the ribose to the glucose. This is what you do. So you have all of these, and you see all these weird sugar names. You turn it into all these different sugar names, and you end up, at the end of it, you end up having glucose come out the other end. This is the way I want you to see it. I want you to see that each of these pathways, just look at the number of carbons in the sugars. And I guess there's one sugar that you know. If you look right in the middle of the paper, you see glyceraldehyde 3-phosphate. That is a 3-carbon sugar, and you know what that is. So what you do is you take 5-carbon sugars, and you take 2 carbons from one sugar and put them on the other one. So you make a 7-carbon and a 3-carbon. Then you take 1 carbon off the 7-carbon and put it back on the 3 one, and you make 6 carbons and 4 carbons. And lo and behold, you've made your first 6-carbon sugar, but you have a 4-carbon left over. So then you react that with a 5-carbon to make a 6-carbon and a 3-carbon, and finally you do that twice, and you end up with 2 3-carbons, and you combine those into a 6. I would never have been able to come up with this myself, but I can just look at that and say, oh, there's 30 carbons in red boxes on the left. There's 30 carbons in 5 boxes on the right. We have rearranged 30 carbons from 5-carbon sugars into 6-carbon sugars. And I just think that's really cool that your cell has figured out how to do it. And I'm willing to sit back and not ask you any details. I want you to know that this is cool. And so that's our next to last mini lecture.